And a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Achieving High Yield and Purity in Lentiviral Production Using dPCR and IEX Membranes. I'm Lauren Coyle, an editor at BioInsights, and joining me today for the presentation is Jay Huang and Oksana Tsygankova, who will address how dual method approach using digital PCR and P24 antigen ELISA enables high sensitivity detection of replication components lentivirus to meet regulatory expectations. After the presentation, we'll have a live Q&A session. So we invite our audience to pose your questions using the Ask a Question box to the bottom of your screen, and we'll try to get to it during the session. Now, I'd like to introduce our presenters. Jay is a senior scientist in process development with over 9 years of expertise in biopharmaceuticals, spanning biologics like mAbs and viral vectors. At SK pharmteco, Jay innovated a novel lentiviral DSP platform, improving vector recovery over twofold and surpassing client expectation. He optimizes diverse downstream processes, including AEX, TFF, and stereo filtration. Oksana is a senior scientist with 2.5 years of experience in GMP testing for gene and cell therapy within the virology department at SK pharmteco. During her tenure, she has played a key role in developing cell-based methods and successfully establishing GMP-compliant assay for detecting replication component viruses and viral vector drug products. So without further ado, I'll hand over to Jay and Oksana to kick off the presentation today.
Hello, everyone. Thank you for joining us today. My name is Jay Huang. I'm a senior scientist process development team at SK pharmteco. So today, Oksana and I will present on achieving high yield and purity in lentiviral vector production, a platform approach using dPCR and anion exchange chromatography membranes. We'll start with a brief introduction of our company, SK pharmteco. Then Oksana will present on dPCR, and we'll end the presentation with our new IEX process for lentiviral vector production. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of the disease. For up-to-date licensing information and product-specific disclaimers, see the respective QIAGEN kit instructions for use or user operator manual. QIAGEN's instructions for use and user manuals are available at www.qiagen.com or can be requested from QIAGEN Technical Services. SK pharmteco is a global multimodal CDMO. We are a key part of the SK Group, the second largest conglomerate in South Korea, and our foundation is built on their strong legacy in advanced materials, life sciences, and technology. This unique position provides us with unparalleled stability and resources, enabling us to be a committed and powerful partner in your journey from small molecules to gene therapies and analytical services. SK pharmteco is a leading CDMO with a strategic global footprint designed to mitigate potential supply chain risks and support your advanced therapy programs. We have a wide array of manufacturing plants, process R&D centers, and analytical service sites located across the US, Europe, and Asia. Our capabilities include dedicated gene therapy facilities in Pennsylvania and just south of Paris, offering comprehensive support for your manufacturing needs. Here's our gene therapy site in King of Prussia, Pennsylvania, located just outside of Philadelphia. This facility serves as a cornerstone of our viral vector capabilities, offering services that span from process development all the way through viral vector production and analytical testing. With our robust offerings, this site is well positioned to support and accelerate your viral vector programs. Our site in France complements our Pennsylvania operations, providing a robust presence in the EU for our clients. We offer comprehensive process development, viral vector manufacturing, and analytical testing, specializing in both AAV and Lenti platforms. With its commercial-ready status and impressive capacity, our France facility is designed to accelerate your gene therapy projects to market. So now I'll hand it over to Oksana to present on dPCR.
Hello, my name is Oksana Tsygankova. I'm a senior scientist in tasting services and analytical development department at SK pharmteco. The title for my talk today is SKPT Testing, dPCR Present and Future. dPCR is an established method in the testing analytical development department. It's used for numerous purposes, including the determination of viral genomic titer, E. coli plasmid copy number, RNA titer, and vector copy number for lentivirus-based vectors. We employ QIAQT platform system for dPCR and RT-PCR. It's a reliable and widely used instrument from QIAGEN. We also buy QIAGEN materials to use along with it. This slide presents an analysis of viral titer and SKPT HEC293 4G9 cells, employing both dPCR and ddPCR. LV titer was analyzed using 5 different probe sets, and each set was run 3 times on QIAQT and once on QX1. As we can see, the results obtained on both instruments are very similar. dPCR was used for clonal selection of SKPT HEC293 cells adapted into growth in serum-free suspension. The cells were cloned using 13 Fc technology that resulted in approximately 100 individual clones. Clones with the best growth properties were selected and tested for LVV production by dPCR. Clone for G9 was chosen, and Jay will discuss it in the next section. The virology department at SKPT developed and validated methods for the detection of replication-competent viruses. The presence of these viruses in vector-based drugs is a threat for patient safety. They can be generated by plasmid recombination during production steps and can undermine the viral drug manufacturing. We use GMP-compliant cell-based assays with PCR endpoints to detect the presence of replication-competent viruses in vector-based drug products. This is an example of cell-based assay for the detection of replication-competent lentiviruses in C8166 cells transduced with HIV-based vectors. The assay starts with the amplification phase that is followed by the indicator phase. The amplification phase consists of a few cell passages, and the last passage is inoculated into fresh indicator cells. After 5-6 days, cells are harvested and analyzed for the presence of RCL by P24 ELISA and GAG-POL PCR. Future developments for these assays relate to the deployment of dPCR. That will eliminate standard curves for RCV assay. dPCR is more precise at low copy numbers and less sensitive to inhibitors than qPCR. It should be easy to switch from qPCR to dPCR. dPCR also generates similar data as ddPCR, but it's faster and simpler and makes a desirable substitute for more complicated ddPCR. We hope that dPCR will enhance the flexibility of our assays and will make them more efficient and cost-effective. I'm going to stop my presentation here. Thank you for letting me to talk about our work at Testing Services and Analytical Development Department. And now Jay will continue his presentation.
Thank you, Oksana. Let's now turn our attention to the lentiviral vector downstream process. We'll begin by discussing some of the key challenges and opportunities we've identified in lentiviral vector downstream processing. Then I'll introduce our new IEX platform process before concluding with the impressive outcomes we've seen from successfully scaling this process from the benchtop all the way up to a larger 50-liter scale. One of the major challenges in lentiviral vector downstream processing from suspension culture is the low overall yield. Even with extensive downstream optimization, the anion exchange chromatography step typically yields only 30 to 40% from clarified harvest, leading to final overall recoveries of just 10 to 15% after sterile filtration. To overcome these limitations and improve downstream yield, a deep understanding and precise optimization of key downstream parameters are essential. To achieve higher lentiviral vector yield in downstream processing, it's essential to first understand the core characteristics and optimize several key parameters, and examine how factors such as the buffer's pH and composition, salt concentration, mechanical stresses like shear and TMP, and time-dependent aspects like hold times and temperature influence the lentiviral vector stability. With this comprehensive understanding, we can then strategically design and fine-tune your downstream process for optimal yield. So next, let's take a look at our SK pharmteco's Lenti downstream platform process. Here's our SK pharmteco's lentiviral vector suspension downstream platform process using our internally developed HEC293-4G9 cell line. Our downstream process takes about 1 to 2 day process from clarification to drug substance, with overall recovery of 20 to 40% from clarified harvest. Our process is very fast and robust and cost-effective plug-and-play downstream process. Our next slide will show you the difference between the traditional anion exchange chromatography step and our optimized anion exchange chromatography platform process. So here on the left, you see our traditional anion exchange chromatography process, which involves a low salt wash to remove impurities, followed by elution and then a strip. In contrast, our new simplified anion exchange chromatography process eliminates the low salt wash step entirely. This innovative design prevents impurity proteins from binding to the anion exchange matrix in the first place, effectively achieving up to 99% whole cell protein removal. This crucial optimization allows more lentiviral vector to bind, significantly increasing the recovery by twofold or even higher, leading to overall lenti yields of minimum 60%. We also compared traditional anion exchange resin and membrane absorbers for lentiviral vector purification. Based on our historical data, we chose membrane absorbers because they're fast and have higher lentiviral vector load capacity, which will reduce the size and the cost of the column. Based on the load capacity, it'll cost about $3,000 with membrane absorbers compared to over $20,000 for resin at the 50-liter scale. First, we tested our current process over traditional process using commercially available anion exchange resin. When we tested these two methods using a pre-packed IEX column, the traditional process yielded only 17%, whereas our improved method achieved a 70% yield, representing a fourfold enhancement using a pre-packed IEX column. While this data was highly promising, we further tested our optimized process using membrane absorbers to reduce overall processing time and the material cost. So we used our optimized AX process using commercially available membrane absorbers and achieved 86% lentiviral particle yield and impressive 110% functional yield, completed in 15 to 20 minute IX processing time from load to strip. As you can see from the chromatogram, our optimized process is very simple. This initial screening result is very promising, and based on this, we further optimized our membrane absorber process, specifically focusing on enhancing both purity and recovery, which I'll show you on the next slide. We further optimized our process with membrane absorbers, and we were able to achieve a 98% whole cell protein removal from the load, while maintaining strong recoveries of 45% lentiviral particle yield and 85% functional yield. These highly promising results led us to further evaluate our method's robustness across various lentiviral vector load concentrations, which you'll see on the next slide. We processed various load concentrations and saw that we consistently achieve high lentivirus yield, ranging from 60 to 110%. This significantly exceeds the industry benchmarks of about 30% yield, confirming its reliability and strong potential for efficient, large-scale manufacturing. So we're now ready to explore its real-world application, which we will focus on scaling up from the benchtop to 50-liter scale. To evaluate the scalability of our Lenti platform process, we conducted runs starting from 2 of the 2 by 2 liter scale and then progresses to a 50 liter scale. For these runs, we utilized our HEC29349 cell line and incorporated client-provided gene of interest. So here's our result from 2 by 2 liter and a 50 liter scale runs. As you can see from this slide, we achieved high IEX step yields of 64% from 2 by 2 liter runs and 77% at the 50 liter scale, exceeding the industry benchmarks of 30% IEX step yield. With this optimized IEX process, we achieved an exceptional 41% overall lentiviral vector yield at the 50 liter scale after sterile filtration, while effectively removing the majority of host cell proteins. The entire downstream processing, from clarification to sterile filtration, was completed in 6 to 8 hours, with downstream material costs just under $20,000 at the 50 liter scale. This performance represents at least twofold or higher improvement over the typical industry benchmarks of 10 to 15% overall lentiviral vector yield, showcasing our platform's capacity for high yield, scalable lentiviral vector manufacturing. In summary, using SK pharmteco's Lenti platform, we've consistently achieved twofold or higher Lenti recovery with up to 99% whole cell protein clearance. With less than 1 hour total IEX time, the entire downstream processing can be performed in 1 day. We've demonstrated superior capacity and consistency, showing at least tenfold binding capacity versus the traditional resin. Our process is highly scalable, successfully transitioning from benchtop to 50 liter scale, making it ready for cost-effective clinical production. Compared to the industry benchmarks of 10 to 15% overall yield, we've achieved an outstanding 41% Lenti yield after sterile filtration at the 50 liter scale using the client-provided gene of interest. Overall, SK pharmteco's Lenti platform offers a clear advantage for achieving accelerated, high-yield, and scalable lentiviral vector production for your specific application. With that, I'd like to thank everyone who joined our webinar today. Please go to QIAGEN.com or scan the QR code on the right to learn more about QIAcuity dPCR system. You may also contact us at skpharmteco.com or scan the QR code on the left to learn how we can support your gene therapy program.
Thank you, Jay and Oksana, for a great presentation there. So we'll now start our Q&A session, so please do keep your questions coming in. So our first question we have here is for Jay, and it is, what specific limitations of resin chromatography beyond processing time and material costs led you to explore the membrane absorbers?
So in terms of the resin chromatography, there are many problems when using this because it has a long residence time of maybe 2 to 4 minute residence time. And also the throughput is very low. You'll be expecting to load about maybe 20 to sometimes 50 mils of the clarified harvest per 1 ml size of the resin. And also for that reason, you will have to increase the column size. So that will also increase the buffer consumptions, and then that will also increase the overall cost of the capture chromatography steps. So by switching this to membrane absorbers, we were able to load the 10 times higher, which is maybe around 200 mils to maybe up to 400 or 500 mils of the clarified harvest per ml size of the membrane absorbers, and then it's very cost effective. And because the capacity is very good with the high throughput, you don't need to use a lot of different buffer volumes. You can also limit the amount of reduce the residence time and then increase the flow rate to get the processing done in less than 1 hour.
And our next question here is also for you, and it's, what challenges have you faced during your IEX optimization?
So in suspension culture, AX step is where the most of the Lenti particle yields typically lost. So you're expecting to see maybe roughly about 30, sometimes 40% recovery from the clarified harvest. And our main challenge is to try to improve the overall lenti recovery, while at the same time without sacrificing impurity clearance, such as whole cell protein. So we carefully optimize several key parameters, including the buffer pH, buffer composition, cell concentrations, and as well as the flow rate. And all of these key parameters, when we optimize them carefully, we were able to, maybe after several rounds of the screening runs, we were able to increase the yield by more than twofold or higher, while at the same time get up to 99% whole cell protein clearance.
Fantastic, thank you so much. And the next question is, how scalable is this integrated platform approach for commercial scale manufacturing, and what are the next steps for expanding its application across different LV programs?
So we've successfully scaled this platform, as you can see from the previous slide, from 2x2 liter benchtop to the 50-liter scale, with consistent recoveries of anywhere from 20 to 40% from clarified harvest, demonstrating that it's ready for clinical and commercial use. Our process is also very flexible, going up from the 50-liter scale to 200 or even 500-liter scale, depending on the client needs. I guess for our next step, we would like to apply this current platform to a variety of lentiviral constructs, supporting for both in vivo and ex vivo applications, and expanding to maybe lenti-like particles. We're also working closely with clients to support tech transfer into their systems.
Wonderful, thank you so much. And what is the maximum shear rate, and what is the range of TMP?
Maximum shear rate of the TMP, and then the shear rate also really depends on how we perform. But usually, if you're going to the shear rate of maybe like 4,000 shear rate or even higher, that will significantly reduce your infectious titer. So our process is a little bit more gentle, going into maybe around like 2,000, maybe anywhere from 1,000 to 2,000 shear rate. This will significantly increase the downstream processing. However, it will also significantly increase the overall infectious and physical yield. So controlling that, and then the maintaining TMP anywhere from 2 to 8 psi should do the trick.
And our next one now is for Oksana. What challenges were encountered during the development and validation of the dPCR assay for LV genomic, RNA, and VSEN, and how were these addressed?
The assay was developed in our analytical development department, and the conditions for this assay were defined so that to get the maximum recovery for RNA and DNA. So the researchers had to determine the concentration for detergent and for time conditions, and also they were working on the conditions for DNase and proteinase K treatments.
And do you get less DNA impurity post IEX?
You do get some clearance from the whole cell DNA after the IEX. A lot of times, lentiviral vectors will eventually co-elute with the whole cell DNA. In our case, we found there's approximately a half log or slightly higher reduction in the whole cell DNA after the IEX. But we are actually very confident because our whole cell protein clearance, the entire downstream process is actually very good. Our TFF step can essentially remove the whole cell DNA process very easily. So we aren't too concerned about that portion.
And Oksana, we have another one for you. What samples can you test for RCL presence?
We can test practically on all steps of manufacturing and production processes, and we offer tests for viral final products, end of production cells, and also vector supernatant.
And this one is for Jay. How do you explain that eliminating the low salt wash with optimized IEX is increasing by twofold the percentage of LV yields?
So in the traditional IEX process, you actually need the low salt wash to remove loosely bound impurity proteins before the elution. The problem with that is you can also elute the lenti during this low salt wash if they're weakly bound to the matrix. So in our optimized process, we improve the key process parameters so that the more impurity proteins, like wholesale proteins, don't bind to the matrix at all. So that would also increases the available binding site for the lentivirus. And also this allows us to potentially eliminate the low salt wash entirely and go straight to the elution, leading to about twofold or higher increase in yield while still maintaining high purity.
Great, thank you. And are there many membrane absorbers on the market for LV, and how did you choose which to try?
There are many different membrane absorbers that you can use. For instance, you can use the Mustang Q, that's one of the popular ones. You can also use Autobind Q. There's also not a membrane absorbers, but things like a SymQA would also work for the lentiviral vector capturing step. These are, in order for me to choose like which one would be the best case, I also try to analyze in terms of the cost. So for instance, if the cost is too high, I try to work on something that is less pricey. And also scalability is very important. So I want to make sure that the one that I'm focusing on is not just a small scale run, such as like 1 ml size, but I want to also try to scale it up to be able to do something in the 50 liter scale or 200 liter scale or 500 liter scale. So those are the key things that I was when it comes to choosing which type of a membrane absorber I should use for the lentiviral vector purification. And then our process itself actually works with not just the one specific type of the membrane absorber, but it actually works on a variety of the membrane absorbers. So from our standpoint, we can easily pick and choose depending on the client needs. If the client is using their, let's say the Mustang Q as their current process, and then if this is for the direct tech transfer run, we can also implement our process over to their Mustang Q and still be able to achieve a very good recovery.
Amazing, thank you. And I think we have time just for one more question. So are there any important considerations for controlling temperature and lentivirus in these processes, or is this mostly circumvented by speeding the process up and making it as fast as possible at room temperature?
That's actually a very good question. So the temperature is one of the key things to consider when it comes to the downstream processes. So so initially we try to process everything one day and hoping to get the best recovery as possible. The slide that we shared today at the 50 liter scale was can also definitely perform in within the 6 to 8 hours. But we also found out later that the process can actually be held for overnight at 2 AC and then processed in the next day. So that will be some benefit for our process and also for GMP teams where they need longer processing time to prepare everything. They can actually put the IEX capture LU8 to their 2 AC overnight and then process the remaining downstream the next day. So that will also help them to, instead of cramming everything up, they can always get the first capture step done, put it on 2 AC overnight the next day, do the remaining process, and can also still be done with a very nice overall recovery.
Well, thank you, Jay and Oksana, for answering those questions, but that is all we have time for today. So any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources tab on the right-hand side of the screen or in the on-demand email. Additionally, an attendance certificate is available on the right-hand side too. So all that's left is to thank Jay and Oksana once more for their great presentation, and thank you to you, our audience, for watching. We'll hope you'll join us again soon.
QIAGEN.
Sample to insight.